Good afternoon, ladies and gentlemen, and my name is Chris Griffiths. I'm currently the president of the ESDR, and welcome to the fourth in our series of ESDR Kitchen live Zoom webinars, which are keeping the ESDR scientific community in touch with the latest developments in skin science, and hopefully we'll find today's one as educational as the previous three. Today it's going to be around Freshly Baked, which is the latest high impact research findings coming straight to you from the ESDR Kitchen. And our co-chairman today are Professor Leo Eckhart from Vienna, who will be Master of Ceremonies for the discussion at the end of these two presentations. But to start off, I'd like to welcome Professor Edel from London, who's going to introduce the first of our two speakers today. Edel.
Thanks, Chris. So the first speaker is Cindy Orvain, who did her PhD on hidradenitis suppurativa at the Institut Mondor in Créteil, near Paris. She's currently doing a postdoc at the Institut Cochin in Paris within the Alanor lab on systemic sclerosis. So she's going to talk about her publication in the JCI on hair follicle stem cell replication stress driving inflammation in hidradenitis. I'd just like to remind everybody before Cindy starts that there will be question and answers at the end of the two presentations. And if you could write your questions and answers in the Q&A or chat on Zoom. So Cindy, we look forward to hearing your talk, and if you could please share your screen. Thanks.
So good afternoon, everybody. So I'm Cindy Orvain, and thanks. First, I want to thank the organizer of the ESDR webinar for allowing me to present my PhD results, which were recently published in Journal of Clinical Investigation. So today I'm going to talk about the hair follicle stem cell role in hidradenitis suppurativa. So hidradenitis suppurativa is a chronic inflammatory dermatosis which appear in hair-rich area, and it causes painful and odorous lesions as abscesses and fistulae, as you can see here on the picture. So it is a common and disabling disease because it affects 1% of French population, and a study has shown that HS patients have the worst life quality among people who suffer from dermatological disease. Unfortunately, there is an incomplete remission of this disease, and available treatment for HS patients are long-term antibiotics, anti-inflammatory molecules, or large excision surgery to remove the lesion. So the common mechanism of hidradenitis suppurativa development is that you have an occlusion of the hair follicle, which will lead to its dilatation and then its rupture in the dermis, initiating the inflammation. It will cause abscesses and then formation of tunnel and fistulae. So the hair follicle seems to be the primum movens of the disease. That's why our team was interested in hair follicle stem cells. So these cells have the ability to renew the hair follicle, and they are located in the outer watches below the sebaceous gland, and they gave rise to the outer watches by proliferation and differentiation. So to study this cell, we collected skin waste surgery. Then we took some piece that we put in dispas, which will allow us to separate the epidermis from the dermis. We collect then each follicle, and we put this follicle in trypsin to collect the follicular keratinocyte. This keratinocyte are then put on culture on irradiated murine fibroblasts, and 3 weeks after the initial seeding, we observe the appearance of first ORS colonies. So our previous paper has shown that we have a spontaneous secretion of two pro-inflammatory cytokines, which are IP10 and CCL5. So my thesis work on the aim of our paper was to characterize the molecular mechanism involved in the pro-inflammatory phenotype of HS-ORS cell. First, we perform a transcriptomic analysis of LC donor on HS-ORS, and we found that we have a deregulation of cell cycle pathway on the type 1 interference signature. We confirmed this type 1 interference signature by studying the RNA expression on total hair follicle cell from LC donor on HS hair follicle. So you can clearly see here that we have an increase of the expression of MX1, IP10, I527 and OAS1, which are 4 interference stimulated genes in HS hair follicle compared to LC donor hair follicle. Then, to confirm the cell cycle deregulation, we decide to assess the ORS cell proliferation rate by doing a colony forming assay. So you could clearly see here that for HS-ORS, we have an increased number of colonies, and the size is bigger compared to LC donor ORS, suggesting that all cells have a higher proliferation rate. So then, to try to understand why we have this increased proliferation rate in HS-ORS, we analyze the hair follicle cell phenotype and their proportion by flow cytometry. So we base our analysis on the paper of Inu-Keta, which exclude the melanocyte which express the CD117 and leukocyte which express the CD45. And then we look at the expression of the marker CD200 and CD34. Cells who express the CD200 only are the bulge cell, which contains the hair follicle stem cell. Cells who express the two markers are the upper subbulge cell, and cells who express the CD34 only are the subbulge cell. Then, in each subpopulation, we look at the quiescent population. So the quiescent population expresses the cytokeratin 15 and the CD49F. So what we obtain? We could clearly see that we have an increase of the subbulge population in HS hair follicle compared to LC donor hair follicle. We also observe a decrease of the bulge population in HS hair follicle compared to LC donor hair follicle. Then, when we look at the quiescent population, which express the cytokeratin 15 and CD49F markers in bulge population, we found that this population is decreased in HS hair follicle compared to LC donor hair follicle. So we could say here that we have a decrease of quiescent bulge population in HS hair follicle in favor of proliferating cell, which is consistent with our previous results. So to go deeper in analysis, we perform a cell cycle analysis, which reveal an accumulation of HS-ORS in ACE phase. When you have an accumulation of cell in ACE phase, it could be due to a replicative stress. So we analyze the replication fork progression by DNA fiber spreading in collaboration with the team of Philippe Passero at Montpellier. So briefly, we incorporated two thymidine analogs, which are the IDU and the CIDU, and then we measured the length of the fiber of IDU. So here you could see that for LC donor ORS, we have approximately the same IDU length, which is around 5 micrometers. But when you look at the HS-ORS, you have an increase of the IDU track length, which is very variable in our cell, suggesting an alteration of the replication fork progression on the replicative stress. So when your cell will encounter replicative stress, it will trigger the DNA damage pathway and especially the checkpoint ATR. ATR will then phosphorylate the effector molecule, which is effector molecule CHK1. So we investigated the presence of phospho-CHK1 on total hair follicle cell of healthy donor on HS hair follicle. So you could clearly see here that we have an increase of the percentage of cell with phospho-CHK1 in HS hair follicle compared to healthy donor hair follicle. And then we notice that cells who express the phospho-CHK1 also express the cytokeratin 15, showing that cells who have an activation of DNA damage pathway are the hair follicle stem cell. So we then pursue our study by studying the micronuclei. So micronuclei are formed when you have a lot of DNA damage. So we stain our cell with lamine B1, which is a marker of the nuclear membrane. And you could see here that we have an increase of the percentage of ORS with micronuclei in HS compared to healthy donor. Then when you have DNA damage, it could lead to release of DNA in the cytosol. So we stain our cell with an anti-single-strand DNA, and we found an accumulation of cytosolic DNA in HS-ORS that we didn't observe in healthy donor ORS. So so at the beginning of my presentation, I told you that we have a type 1 interference signature. And when you have accumulation of cytosolic DNA, it leads to activation of sensing, of DNA sensing pathway. And in this case, this is STING, which is activated. So STING will then induce the transcription of type 1 interferon and interferon stimulated genes. So to see if STING is involved in our type 1 interferon signature, we transfected our cell with scRNA directed against STING, and we studied then the expression of type 1 interferon and interferon stimulated genes. So you could see here that we have a decrease of the expression of interferon beta, OAS1 and IP10 in HS-ORS, which are transfected with the scRNA against STING, suggesting that STING is involved in our type 1 interferon signature. We performed the same experiment with a scRNA directed against I516, which is also a DNA sensor, and we found that we have a decrease of the expression of interferon beta, OAS1 and IP10 in 4 HS-ORS transfected, suggesting that I516 has also a role in our type 1 interferon signature. So to conclude, we propose here a new model for the HS physiopathology. We have an increase of the proliferation rate of the bulge cell, which will lead to a replicative stress of this cell, formation of micronuclei and release of single-strand DNA, which will then be sensed by I516 and STING. STING will then activate TRS3, which will lead to the production of type 1 interferon and interferon stimulated gene like IP10, which will initiate the inflammation in hidradenitis suppurativa. So I would like to thank the team where I perform my PhD, so my thesis director, Sophie Hu, and the engineer who work daily with me, Francette Jean-Louis. I also thank the dermatology and plastic surgery department of Henri Mondor Hospital who give us the skin explant, and the Human Genetic Institute of Montpellier who help us a lot on DNA damage and replicative stress, and of course HS patients for agreeing to be in part of our study. and the sensory intention.
So thank you very much, Cindy, for that fascinating talk. And please hold on for the questions and answers at the end. I'm sure lots of people will have lots of questions for you. So very pleased to introduce the second speaker today, Professor Wim Declercq, who's a Professor of Molecular Biology at the University of Ghent. He is a well-known attendee at the ESDR and has been active in the field of TNF receptors, caspases, cell death, keratinocyte differentiation, skin cancer, and inflammation for many years. His lab currently focuses on studying the involvement of the RIP kinases in skin homeostasis and pathology. And he's going to talk today about his recent publication in the Journal of Experimental Medicine about sensing of endogenous nucleic acids by ZP1 induces necroptosis and skin inflammation. So thank you, Wim. If you could share your slides.
Okay, thanks for the nice introduction, and I also want to thank ESDR for giving me the opportunity to present our recent paper on sensing of endogenous nucleic acids by ZBP1, which in certain conditions can induce cell death, necroptotic cell death known as necroptosis, and can cause skin inflammation. So as been told by the previous speaker already, so there are a number of nucleic acid sensors known in the cell. And maybe the best known to the general public are the TLRs, the toll-like receptors, which can sense different types of endogenous or let's say microbial nucleic acids. Now, these are membrane associated, but you also have a number of purely cytosolic sensors like MDA5, RIC1, STING, which you just heard about, but also ZBP1, which stands for zDNA binding protein 1, or in the past it was often called DIE. Now, although it has been originally characterized as a zDNA binding protein, it's now more and more data show that it's in fact an RNA sensor, and probably a double-stranded RNA sensor. Now, all these sensors, they initiate signaling cascades, as you heard also in the previous talk, and many of these signaling cascades can lead to inflammation, so causing then probably inflammatory diseases in certain organs. Now, ZBP1 originally characterized as a sensor of viral nucleic acids. So there are a number of viruses which have been shown to activate ZBP1, for example, influenza, the Zika. So you have vaccinia. So you have a number of viruses which can produce nucleic acids, which then are interacting with the two nucleic acid sensing domains in the N-terminal domain, and this can ignite signaling cascades. Now, ZBP1 can also, because of its RIP interacting homology motif, can interact with RIP kinases. For example, with RIPK3, which can then nucleate to a RIPK1, RIPK3 signaling complex, which can activate gene activation, but also activate cell death, for example, apoptosis, but also in certain conditions, necroptosis. Necroptosis occurs when RIPK3 phosphorylates MLKL, which is a pore-forming protein, which then oligomerizes and permeabilizes the cellular membrane and gives rise to necrotic cell death or cell accompanied by cell rupture. Now, these RIPK1 and RIPK3 proteins are signal transducers. They are best known for their signal transduction in stimulated by death receptors. For example, TNF receptor is mainly studied in that context, and it can activate genes and also cell death, as I just mentioned. Now, several years ago, by work from many labs, also our lab, it has been shown that RIPK1, in fact, has two different activities. So it has a kinase-dependent function, and I already alluded to the fact that it can induce apoptotic cell death as well as necroptotic cell death, and that depends on the cellular context. But it also has a kind of scaffold function. So in its inactive status, in its kinase death status, it prevents sensitization of cells to cell death. And depending on the tissue in which you ablate RIPK1, it can sensitize to RIPK3 activation and necroptotic cell death, for example, in the skin, or it can ignite caspase cascades and result in apoptotic cell death, like, for example, in the gut. And this has been shown by many different labs all over the world, including ours. Now, coming back to RIPK1 ablation in keratinocytes, so here you see the control mice. So these are the kinase dead mice, knock-in mice. So they don't develop any pathology. They have no significant phenotype, spontaneous phenotype. But if you ablate RIPK1 in the keratinocyte compartment by K5 or K14 driven deletion, then these mice will develop skin lesions and inflammation. And this is something we have shown a few years ago already, so that this inflammation is really due to RIPK3 dependent necroptosis. So here you see the RIPK1 eco mice. So after 4 or 5 weeks, they will develop spontaneous lesions, which really aggravate, and you have to euthanize the mice for ethical reasons. But if you cross these mice, for example, with homozygous RIPK3 deficient mice, then you can see they are largely rescued to develop skin lesions. So they start developing skin lesions only after 20 weeks, and the skin lesions are mild. So these mice breathe normally and so on. So they don't suffer too much. You can keep them at least for a year, let's say. Now, a few years ago, it was shown by 2 independent groups, one of Manolis Pasparakis and the other one led by Vishwa Dixit, that this RIPK3 mediated necroptosis in the absence of RIPK1 is driven also by ZBP1. So if you delete ZBP1, you can also protect these mice against developing skin inflammation or inflammation in other tissues when it concerns necroptotic cell death. Now, one of the major questions is, of course, is nucleic acid sensing required for the ZBP1 dependent cell death induction and development of skin lesions in these RIPK1 eco mice? Remember that ZBP1 was till very recent only considered to sense viral nucleic acids. So therefore, we used sensing that ZBP1 knock-in mouse model. So this is a schematic presentation of ZBP1. You have the 2 nucleic acid sensing domains, and these were disrupted by 2 point mutations in each domain, and then knock-in mice were developed. And this is just a sequence analysis to show that these mice indeed carry the correct mutations which ablate nucleic acid sensing by ZBP1. Now, the skin pathology of RIPK1 eco mice, to our surprise, was dependent on this nucleic acid sensing by ZBP1. So here you have again the RIPK1 eco mice, and when you cross them with homozygous ZBP1 mutant mice, so which have lost their sensing capacity, you can see that they are largely rescued. They are not completely rescued, so they still develop lesions, but these lesions again are also mild. So this indicates there is still something else going on in these mice, which can also lead to kind of milder lesions. This Western blot here shows that ZBP1, the ZBP1 mutant is undergoes normal regulation in these keratinocytes. So we isolated keratinocytes, and the ZBP1 is an interferon stimulated gene. And when you treat the keratinocytes with interferon, you can see upregulation of ZBP1 in the wild type situation. And this is also the case for the mutant, and it's giving similar levels of ZBP1 in both genotypes. And this Western blot confirms that indeed RIPK1 is completely ablated in these keratinocytes. Now, if you look at the histological levels, if you look in these lesions which lack RIPK1, then you can see there's a huge thickening, epidermal thickening, hyperproliferation is going on. You also have aberrant expression of, for example, inflammatory keratins like K6. We also have done other markers, so people who are interested in that should look in the recent paper. Now, when you rescue them with crossing with the ZBP1 mutant mice, you see that that there are no lesions and K6 expression is restored to normal, so only in the hair follicles. So this ZBP1 activity also drives an IL-17 immune response in RIPK1 eco mice. So I don't show all data. There are some additional data in the paper. I can't show all data because of time constraints, but as you can see here, there are a number of immune cells which infiltrate into the epidermis. So the CD4 cells are increased, NKT cells, neutrophils, CD64 positive macrophages are also infiltrating. And we also did cytokine stainings, and we show, for example, that the CD4 T cells are largely consisting of IL-17 producing T cells, so they are probably Th17 cells. So they are negative for interferon and positive for IL-17. This is something you can also find in the paper. Now, if this whole phenotype is really depending on ZBP1 activity in the keratinocytes, then that would be logic that ZBP1 is also mainly expressed in the keratinocytes. And this is something we checked, for example, with immunohistochemistry. So we stained for ZBP1, and here you can see that ZBP1 is mainly expressed in the keratinocytes. And if you zoom in a bit closer, you would see that it's a cytosolic ZBP1 expression. This is also confirmed at the messenger RNA level. There are some other interferon signatures, which are genes which are upregulated. Interferon gamma is also upregulated, and some other inflammatory parameters which is kind of normal in this type of skin inflammatory pathologies. Now, to show that it's really cell autonomous, then you would expect that if you prevent cell death just in the keratinocyte compartment, that you would rescue the phenotype, and this is the case. So if you have the RIPK1 eco mice, so again, these are the control mice which develop very rapidly these skin lesions. When you cross them with MLKL eco mice, so only deficient in the keratinocyte compartment, again, you see this rescue phenotype. And there is some other pathway going on, which is seemingly then MLKL independent. Now, to elaborate on that, we isolated keratinocytes from the different genotypes, and then we stimulated them with interferon. So interferon will result in ZBP1 upregulation. And then the question is this sufficient to induce cell death? And the answer is yes, but only in the ZBP1 wild type expressing genotype. So if you have keratinocytes which carry the ZBP1 sensing that mutant, you see that in these cells, interferon cannot induce cell death, meaning that it's sensing sensitive ZBP1 signaling. We also took two controls along, TNF, which hardly affects RIPK1 wild type or RIPK1 eco keratinocytes, poly-IC, which is a TLR3 stimulator, can induce some cell death, but this is independent of the ZBP1 genotype. So this was further confirmed by experiments in which we introduced ZBP1 wild type and ZBP1 mutant by means of lentiviral overexpression in primary mouse keratinocytes. So in the RIPK1 wild type situation, nothing happens, but when they are sensitized, so in the RIPK1 ablated situation, you can see that again, the cell death is dependent on the wild type situation and not, so the ZBP1 sensing that mutant is not able to stimulate significant cell death. And this is just here a Western blot to show that indeed these proteins are expressed in the correct way. So what is the take-home message? I hope I convinced you that these data indicate, like also many other people have been showing now very recently, that ZBP1 can also be activated by endogenous nucleic acids. And in a few other papers which are mentioned here, so they provide evidence that it's probably based on retroviral double-stranded RNA sensing. The ZBP1 activation can then ignite cell death pathways leading to necroptosis and tissue inflammation. In these RIPK1 ecomice, there are also other pathways going on, so probably TLR3, which can also result in RIPK3 activation. However, there are still many questions, of course. So what is the real nature of the ligand of ZBP1? That's not clear. You know, ZBP1 should be expressed, and this is interferon dependent. So what is the trigger, the initial trigger resulting in the interferon, which then leads to ZBP1 expression? How is ZBP1 exactly activated? In some papers, they claim that there is a nuclear activation. Others say it's cytosolic activation. So this should be clarified, let's say, in the coming years. I have some people to acknowledge. So Michael de Vos and Giel Tange were shared first authors on this paper. Jonathan Malfait is my sharing senior author. And I thank Jan Reewinkel for sharing his mice to do these studies. And I thank you for listening, and I will take questions.
Thanks very much, Wim, for a very interesting talk. I'll pass over to Leo now, who's going to moderate the questions and answers.
Yes, thank you very much to Cindy and Wim for the great presentations. There are a number of questions, and I tried to read many of them, and please try to give brief answers so that we can address most of these questions. So the first question goes to Cindy. So are there similar mechanisms at play in the hair follicle in other inflammatory diseases? And associated with that question, are the type 1 interferons increased systemically in the HS patients? So 2 questions.
So for my knowledge, there is no similar mechanism in other follicle inflammatory disease because, for example, in alopecia areata, it is progenitor cells which are affected and not hair follicle stem cells. In scarring alopecia, this is destruction of hair follicle stem cells, but not a loss of quiescence. And the last one for androgenetic alopecia, this is a lack of conversion of hair follicle stem cell into progenitor cells. And for the second part of the question, we didn't observe type 1 interferon for all HS patients because this is there is a huge heterogeneity. And this is, we have only a group of patients who will express with high level interferon, type 1 interferon.
Okay. Okay, thank you. Another question from, again, to Cindy, from Daniel Johnston. He's wondering why you did not investigate phosphosting, but only sting itself.
Yes, it's an interesting question indeed. Normally, we should do it to see, but I guess we didn't have time. It was the end of my PhD, so I cannot. But it's a good experiment to do. This I wanted to do, but then I don't have time, and I do other experiments and other stuff.
Okay, thank you. So now let's also ask a question to Wim. So there was one question about the microbiome. Is it involved in this in this model? Have you tested everything under germ-free conditions?
Well, these experiments are still running because, yeah, of course, you have some barrier disruption in these mice, and it could be that the microbiome contributes. Now, from the data we have currently, it's not really rescuing the appearance of the skin lesions. So in germ-free conditions, the skin lesions still appear. After 5 weeks, you see them appearing. Although we have the impression for the mice that develop it, they are a bit milder, which is not so unlogic because, yeah, probably it contributes by amplifying once there is an inflammatory problem. Let's say the microbiome may amplify it, but it's not protected. And this is a bit in, let's say, in agreement with another study, a recent study, one of the Nature papers also, where they showed that ZBP1 can also be involved in, for example, in gut inflammatory conditions, in an auto-inflammatory disease, which is due to chromatin modification. And there they also did, in these mice, they also did antibiotics experiment. And also there, it had a mild effect, but no dramatic effect on the development of the pathology.
May I ask about an alternative hypothesis? So about the retroviral also nucleic acids. So if this is also involved, I mean, you mentioned it, would it be in the differentiated keratinocytes or in the proliferating keratinocytes where this signaling is initiated?
Well, we didn't exactly do these experiments, but Manolis Pasparakis, they did some RNA sequencing experiments. And what they see there, that these retroviral elements are expressed both in wild type as in RIPK1 deficient keratinocytes, more or less at the same level. Of course, once you have inflammation, then it's in one condition and not in the other. It's not surprising, of course, that also these expression levels go up. So there are, it's a bit an analogy with other senses, like for example, MDA5, which is also involved in autoimmune diseases, where it's shown that it binds these allo repeats in humans. There are some indications that this will also be the case for ZBP1, mainly these small interspaced repetitive elements. I think it makes sense.
Okay, thank you. So another question for Cindy. So what is driving the proliferation of the outer root sheath cells?
Yes, so I think I could say that this is a question. This is what my team tried to understand. So we do some investigation with exome analysis. So to see if we can explain this with some genetics. We also